Hey everyone, welcome back. And today I'm going to start off by just kind of summarizing the events leading to and following an action potential. So then again, this is the electrical activity in a neuron that makes up the basis for the communication between neurons and other cells. So again, it's all about establishing an electrical current in that neuron. And electricity, again, is just movement of charged ions. In this case, it's going to be movement of positive ions, and that's going to change the voltage across the membrane of the neuron. So here in this green line, I've kind of drawn a membrane with the relevant channels, ion channels that are opening and closing that cause this electrical activity. And here I have a graph of how the voltage changes over time, with this little dotted line representing the threshold value, past which you get the formation of the action potential. So again, the resting membrane potential, or the resting voltage across the membrane of a neuron, is going to be about -70. So this is a neuron at rest that has not yet been stimulated. And that stimulation is going to be based again on the binding of a ligand, like a neurotransmitter, or some mechanically sensitive channels opening. And those stimuli are going to cause positive ions to these gates open up, and positive sodium ions are going to rush into the cell, because the cell is negative on the inside because of the activity of those sodium-potassium pumps and the leak channels. But so the neuron gets stimulated at the dendrites, or the cell body, and that stimulation again is because of these mechanically and ligand-gated sodium channels, and they're going to open in response to that stimulus. And this is going to cause some depolarization. So the cell is normally polarized. It's got positive on the outside, negative on the inside. And when you allow positive ions to flow inside, that raises the voltage. It's going to depolarize that neuron. And each time you get some stimulation, that's called a graded potential. So sometimes there's just a weak stimulation, and it doesn't allow the neuron to reach the threshold. And so if there's no further stimulations, it'll go back to the resting membrane potential. Now, if there is significant enough stimulation, or enough a high enough strength of stimulation, or enough frequency of stimulation, opening enough of these sodium channels to get the voltage up to the threshold, that's when you get voltage-gated sodium channels at the trigger zone of the neuron starting to open. So you reach that threshold, and then these voltage-gated sodium channels are going to open along the length of the axon, and that causes the voltage to rapidly depolarize. The cell rapidly depolarizes. The electrical signal is going to propagate or shoot down the axon, and ultimately cause the release of neurotransmitter, or the stimulation of the next cell in the chain. So this increase, this depolarization, that is the action potential. So after that action potential occurs and the cell is now depolarized, these voltage-gated channels are going to start to close. And actually, during the action potential itself, you get voltage-gated potassium channels that are starting to open. So remember, sodium is the positive ion that's outside of a neuron. And potassium is also a positive ion, but those pumps work to pump potassium into the neuron. So the neuron has a lot of positive potassium ions in it. Not enough to make it a positive voltage inside, but it does have positive potassium ions inside it. The reason for the cell storing these potassium ions is so that it can release them and bring the voltage back down. So you get voltage-gated potassium ion channels opening after the action potential. And those potassium ions that were inside are going to flow outside with their concentration gradient. Because there's so many of them inside the cell, they want to escape. And potassium ions, positive ions leaving the cell, is going to repolarize the neuron. So in allowing all these positive potassium ions to now leave the neuron, that is going to bring the voltage back down to a negative value, and that is called repolarization. So you're reestablishing that negative resting membrane potential. So enough positive ions flow out of the neuron actually that it's going to hyperpolarize the neuron. For a brief period of time, the voltage is going to be below the resting membrane potential. So at rest, the resting membrane potential is minus 70 millivolts. And after an action potential, that depolarization is going to actually occur until the neuron is hyperpolarized. So the voltage is below minus 70. But that's going to balance itself back out. Those sodium-potassium pumps and the potassium leak channels are going to bring that voltage back to the resting membrane potential so that it can be stimulated for producing future action potentials. So now the neuron's ready to fire again. So this is the whole cycle of events that a neuron goes through before and after an action potential. And neurons, some neurons need to fire repeatedly. So this process occurs, in a few milliseconds, a few thousandths of a second. So this happens really quickly. And it's also important to note that during this process, specifically during the process of the action potential itself, and the depolarization and the repolarization, that's going to be what's a period called the absolute refractory period. So during this period, when you've got those voltage-gated sodium channels opening and then the voltage-gated potassium channels opening, the action potential, this is the period of the action potential. During that time, what absolute refractory period means is that during this period, any additional stimulation is not going to cause an action potential to occur again. So once this process has been initiated, no more action potentials are going to occur, no matter how much stimulation is going on in that neuron. So only one action potential happens at a time. Now once the cell has repolarized, actually drew my line a little bit off, so that's going to, the absolute refractory period is going to last up until you get back below the threshold. Now after you've gotten below the threshold, and before you've reached the resting membrane potential, you get a second period called the relative refractory period. Now during the relative refractory period, it's just going to take a lot of additional stimulus to get another action potential to fire, more than it would normally take. And that's because these voltage-gated potassium channels are open. So the more positive ions that come in, that's just going to force more positive ions out. And it's going to be really difficult to bring this back up to the threshold during this period to initiate another action potential. So during these two periods, this one, it's impossible to initiate another action potential by stimulating the neuron. and during the relative refractory period. It's not impossible, but it's much more difficult to bring this value up to the threshold to initiate another action potential. But after the relative refractory period, the neuron is back to its resting membrane potential and ready for another action potential to fire, just like it was at the beginning of this cycle. So make sure you've got a good grip on what the events are that are happening during electrical activity in a neuron, and what the voltage across the membrane of that neuron is going to look like during these events, up to and during and after an action potential. So now that you know about the signaling that occurs after a neuron is stimulated, that's going to result in the generation of an action potential, and that's transmitted down the length of a neuron. Really, the point of all this, again, is communication between cells. So some cells communicate with hormones or chemical signals, and neurons transmit electrical signals. But again, it's just a way for two cells to communicate. And the neuron is communicating with cells like muscle cells, telling them when to contract, or can be communicating with a gland or another neuron, as is the case in the brain. There's connections of many neurons. So this is basically a chain of cells, and they're communicating with one another. And that communication starts with this electrical signal. But where the actual interaction between these two cells happens is at a place called the synapse. And whenever there's a synapse, that means it's the place where a neuron and the cell it's communicating with are meeting. So the parts of the synapse are the presynaptic neuron. You write that in a marker that works. So you have the presynaptic neuron, or the presynaptic cell. And so if this is the synapse, this neuron occurs before the synapse, so it's presynaptic. And this, whatever is receiving the signal, would be postsynaptic. And between those two cells, you have the synaptic cleft, in the case of if this is a chemical synapse. But there's a couple of different types of synapse. One that I just mentioned is a chemical synapse. And a chemical synapse is going to include the release of chemicals, neurotransmitters, from the presynaptic neuron. So the presynaptic neuron, an action potential fires, and that causes the release of neurotransmitters. And that's the basis of a chemical synapse. Those neurotransmitters will bind receptors on the postsynaptic cell and stimulate it somehow. So that's how a message is sent through a chemical synapse. Now the other type of synapse, so we have chemical as type 1, and that's the predominant type of synapse. Most synapses are going to be chemical synapses, where there's a neurotransmitter that acts as kind of the intermediate between these two cells. The other type of synapse would be an electrical synapse. And in an electrical synapse, instead of a neurotransmitter being released as a result of the action potential, these two cells are actually going to be in contact with one another. And there will be attachment proteins that attach these two cells to one another. And there are going to be this junction between these two cells, and there will be channels that are open between these two cells that allow ions to flow between them. So as opposed to this cell releasing a neurotransmitter, the neurotransmitter binds a receptor, that receptor causes ions to flow into the cell, stimulating it. there's no chemical intermediate. The signal, the ions flowing down this neuron are going to travel directly into the postsynaptic cell. So there's no need for neurotransmitter in the case of an electrical synapse. And an electrical synapse occurs in the brain and places that need to process information quickly. And electrical synapses also serve other tissue types, like in smooth muscle, where you have coordination between cells that's rapid, so that they can all be contracting at once. Those cells will be joined by an electrical synapse. But the electrical synapses are going to be more rare in the nervous system than the chemical synapses. So most of the time you're actually going to see neurotransmitter release from this presynaptic cell, and that neurotransmitter stimulates the receiving cell or the postsynaptic cell. So let's walk through the events that happen at a chemical synapse now that lead to eventually what's called a postsynaptic potential. And this is just whatever the effect on the postsynaptic cell is going to be. It's called the postsynaptic potential. And what that's going to do is either allow positive ions to flow into the cell that's receiving the signal, and that would be an excitatory thing. If you depolarize the cell by allowing positive ions in, that's exciting this second cell in the chain. It can also be an inhibitory effect if the result is for positive ions to leave the cell, or to hyperpolarize, or to cause negative ions to flow into the cell. So if you're thinking about the voltage of that receiving cell, it can either be depolarized and go up, or it can be hyperpolarized and become more negative. So this can either, in either case, if you depolarize that cell, that's going to be exciting this cell if you raise the voltage. And if you hyperpolarize it, it's going to basically dampen any excitement of that cell. It's going to inhibit this cell from becoming excited. So these postsynaptic potentials can either be excitatory, basically stimulating it to either send a signal or to contract, or they can inhibit that excitation. So excitatory, or EPSPs, or inhibitory, or IPSPs. Those are the two types of basically the result of communication between these two cells, is either inhibiting or exciting this cell. But so let's look at the events that lead to this communication first. So we've talked about the action potential, how it forms and is transmitted along the axon. So once that voltage increases down here near the synaptic terminal, so this little bulb at the end of the neuron, that increase in the voltage down here is going to trigger a different type of channel, ion channel. And this is a voltage-gated calcium channel. So there's calcium ions flowing around outside the synaptic terminal. And these voltage-gated calcium channels will open in a response to an increase in the voltage here at the terminal. So an action potential is going to bring that increase in voltage. So the voltage increases here. That causes calcium channels to open, and calcium flows into the cell. Another thing is, for this chemical synapse, it's dependent on neurotransmitter release. And the synaptic terminal has vesicles called synaptic vesicles that contain neurotransmitter. So I've drawn these vesicles, these little membrane-bound compartments that store neurotransmitter in the synaptic terminal. And those little sacs of neurotransmitter are only going to release their contents out into the synaptic cleft, which is this space here. They're only going to be released into the synaptic cleft when calcium is present. So calcium flows into the cell, and what that does is causes these synaptic vesicles to fuse with the plasma membrane. So they're going to kind of move to the plasma membrane of the neuron, and they're going to fuse with that plasma membrane and release their contents into the synaptic cleft. So again, action potential, opening of calcium, voltage-gated calcium channels, and synaptic vesicles fusing with the plasma membrane, releasing their stored neurotransmitters. And these neurotransmitters are going to act as ligands. They're going to bind to receptors on the postsynaptic cell. And depending on the nature of these receptors, the effect can either raise the voltage and be excitatory. It can excite this postsynaptic cell, cause it to contract or to release hormones if it's a gland, etc. Or this receptor could be, it could be the case that when this particular neurotransmitter binds the receptor, it's going to cause hyperpolarization. It's going to cause a decrease in the voltage and make it harder to stimulate this cell. So it's going to have an inhibitory effect. So this is how most transmission between neurons and their postsynaptic cells, the postsynaptic targets, is going to occur. Is action potential, release of neurotransmitter, and then you see the effect of that neurotransmitter at the synapse on the postsynaptic cell. Let's take a look now at a closer look at the effects that excitatory postsynaptic potentials would have versus inhibitory. So let's say we have some cell X here. We'll say it's a muscle cell, for example. And it has to reach a certain threshold in order to be activated. So we have here, I've drawn two neurons, and one of them is going to have an excitatory effect on the muscle, and the other is going to have an inhibitory effect. So, and we'll also take a closer look at what exactly is causing that excitation or inhibition. So let's say an action potential travels down this excitatory neuron and causes release of some neurotransmitter. That neurotransmitter binds to a receptor, and that receptor allows positive ions to flow into the cell. So that is going to raise the voltage of this cell closer to that threshold where it is going to have an effect. If this is a neuron, that means we're going to be, with these EPSPs, we're going to be bringing it closer to activating an action potential in this neuron, in the postsynaptic neuron. So that's the effect of EPSPs, and they can add up on one another. So if there are multiple stimuli from this excitatory neuron, we can possibly get it to reach the threshold where that action potential occurs. So at the same time, let's say there's an inhibitory neuron here, and an action potential travels down it, causes neurotransmitter release here, maybe a different neurotransmitter, or there's different receptors here. So the neurotransmitter binds, and it's going to have an inhibitory effect. So what do we mean by that? Well, an excitatory effect is positive going in, raising the voltage. We have to lower the voltage, make it more negative, hyperpolarize it. to have an inhibitory effect. So how do we do that? Well, one way is by opening a channel that allows positive ions to flow out of the cell. So this would be like opening a potassium channel. Would have an inhibitory effect that would lower the voltage away from the threshold value of minus 55. So one other way that we could make it a more negative value is not only to remove positive ions, but to allow negative ions, something like a chloride ion, to go into the cell. That would also lower the voltage, hyperpolarizing it, bringing it further away from this threshold. And if you add the effects, let's say these have equal strengths. So on one side you have an excitatory potential that's happening, on the other you have an inhibitory potential that's happening. Well, these are going to add up on one another, and you're going to get no net change in the voltage because you have positive ions flowing in, but you also have either positive ions flowing out or negative ions flowing in that are going to counter the effects of that excitation. So in order to really excite the postsynaptic cell, you have to have more excitation than you have inhibition going on. And inhibition has its place in the nervous system as well. So at certain times, let's say you want to have some muscle movement or some movement from a group of muscles, and you want to excite those muscles that are responsible for that movement. Well, as you will learn in lab next semester, there are muscles that oppose the movement of other muscles that need to be inhibited at the same time as the other ones are being stimulated. So inhibiting certain cells is going to be an important function of the nervous system, just like exciting other cells. And again, in order to excite a cell, you're going to have to overcome any inhibitory effects and get up to that threshold value. So just a minute ago, I alluded to the fact that stimuli from the presynaptic neuron add up on one another in the postsynaptic neuron. So if you get an excitatory stimulus at the same time as an inhibitory stimulus, those cancel each other out. They add up on one another and cancel one another out. And this is called summation. This is the adding up of all the stimuli to determine what's going to happen at this cell. And there are a couple of different types of summation that we talk about when we refer to adding up the stimuli in these different cells. So let's imagine that we have this one presynaptic neuron, and it's sending excitatory signals to this postsynaptic cell. So let's say it fires an action potential. We'll say action potential number 1. That's going to increase the voltage, since it's an excitatory potential, increase the voltage a little bit toward the threshold. So we would say that's maybe a weaker potential, a weak stimulus. And if there's no further stimulus, that cell is going to come back down to its resting potential eventually. That stimulus will fade. So in order to stimulate this cell completely, to get it up to the threshold, you may need to have numerous stimuli in order to reach this threshold. So what that means is you're either going to have multiple neurons stimulating at the same time, or this one presynaptic neuron is going to have to fire repeatedly. So that leads us to a discussion of the different types of summation. So the first one would be, let's say, instead of just one action potential leading to an excitatory stimulus, let's say this presynaptic neuron is firing rapidly. So there's one action potential, one neurotransmitter release. And again, if there was no other stimulus, you would return to resting for the postsynaptic neuron. But let's say it's firing rapidly. Those stimuli are going to build up on one another. There's not enough time in between stimulations for the postsynaptic cell to return to its resting membrane potential. So if you get repeated firing of this presynaptic neuron, that's those stimuli. Here's the first action potential, here's the second, and then maybe a third is the one that is going to bring it up to the threshold. So if you have one neuron that's repeatedly sending a signal, those stimuli adding up on one another are called temporal summation. So temporal means time. It means this summation is related to the frequency of stimulus. So the more frequently this neuron is sending an excitatory signal, those stimuli are going to add up on one another in what's called temporal summation. Now the other type of summation is called spatial summation. And most postsynaptic cells that are under the control of a neuron are actually going to have potentially many neurons exerting an influence on them. So, for one muscle cell, one muscle fiber, there's potential that there's many neurons that are making a synapse with that cell. And if they're all firing, that's going to be called spatial summation. Is if you have many neurons all giving a stimulatory effect. So temporal summation, again, is just related to the frequency of stimulation. Spatial summation is related to the number of neurons that are sending these excitatory signals. And they don't have to be excitatory. In this example, the stimuli are excitatory, but spatial and temporal summation is going to include inhibitory stimuli as well. But just for this example, we're just thinking about it in terms of the signals adding up on one another being excitatory. Let's talk about now how synaptic transmission comes to an end. So, a synaptic transmission is based, at least for the chemical synapse, on neurotransmitter release from the presynaptic neuron. So action potential happens, release of neurotransmitter, and that neurotransmitter exerts an effect, stimulatory or inhibitory, on the postsynaptic neuron. But how does that effect come to an end? How does that transmission end? And it's based on removing neurotransmitter from the synaptic cleft, this space between the two cells. So in order to stop the transmission, you need to get rid of this neurotransmitter. And that can happen in three different ways. The first is just by diffusion. This is the simplest mechanism for removing neurotransmitter. And that's simply where neurotransmitter just kind of diffuses out. Since it's highly concentrated here, it's going to move to a place where it's less concentrated, and that happens by simple diffusion. And eventually it might get reabsorbed by or absorbed by some other cell, some other neuron that's in the vicinity. So diffusion and reabsorption or absorption is one way to remove neurotransmitter. The next way is degradation. And this is going to be a function of enzymes. So there are enzymes, one of them, a commonly cited example is acetylcholinesterase. So acetylcholine is one type of neurotransmitter, and acetylcholinesterase is an enzyme that degrades acetylcholine. So that enzyme will be floating around in this synaptic cleft, and it's going to destroy or degrade whatever neurotransmitter is in the synaptic cleft. So that gets rid of the neurotransmitter, and that's also going to stop the synaptic transmission. And the last way is reuptake. So neurons, like many cells, they have ways to recycle things, and so it's not just one-way movement of neurotransmitters at the synapse, but neurotransmitter can be taken back up into the neuron that released it. That's called reuptake. And you've probably heard of SSRIs. It's a medication that blocks reuptake for the neurotransmitter serotonin. Selective serotonin reuptake inhibitors is what an SSRI is. And basically the reason for the use of that drug is that you want to exert the effect of serotonin. It's got a pleasurable stimulation that many people, for whatever reason, don't have enough of. And so to keep the effects of serotonin around longer, make you less depressed or whatever it's being prescribed for. If you block reuptake of serotonin, the effect of serotonin persists, and the beneficial effects of it being around persist. So reuptake is the final way where the presynaptic neuron basically vacuums up all of the neurotransmitter that it had previously released. So that's how you end a synaptic transmission. So I would like you to know some general information about specific types of neurotransmitters. And there's a really good summary of this information in your book in Chapter 11, Table 11.3. This is on page 414. And basically what I want you to know about the different types of neurotransmitters is, first, what are they made from? So there are types of neurotransmitters that the precursor is amino acids, for example. That's a large number of neurotransmitters are made from amino acids. Others, like acetylcholine, is made from acetyl-CoA joined with the molecule choline, hence the name acetylcholine. So I'd like you to know generally what the neurotransmitters are, what they're made from, and what's their effect. So almost all of the neurotransmitters are going to have an excitatory effect primarily. Almost all neurotransmitters can have either excitatory or inhibitory effects. But for each one, there's usually a primary effect, whether that be inhibitory or excitatory. And all but GABA and glycine are going to have excitatory effects. GABA and glycine have an inhibitory effect, and everyone else is excitatory. And of course, this is the effect on the postsynaptic cell. And then the last thing I would like you to know about the neurotransmitters in particular is, are they going to be used in the central nervous system, the peripheral nervous system, or both? So many of them are going to be both, but some are just either or. And so I would like you to take a look at that information as well. And that should sum up basically all the information that I would like you to know about neurotransmitters. When we were looking at the different potentials that could occur at a postsynaptic cell, I kind of alluded to the fact that multiple neurons can make contact with the same cell, and the opposite of that is also true. A single neuron can make contact with multiple cells, and this brings us to the concept of what's called a neural or neuronal network. I would say probably or a neural circuit. Neuronal circuit works too, but this is a circuit is just a chain of neurons, And there's a couple of different functional types of groups of neurons when we're talking about these neural circuits. So the first one that I've drawn here is called a diverging circuit, where you have one neuron that is stimulating two neurons. Each of these neurons is stimulating two neurons, and those neurons are each stimulating, in this case, a muscle fiber. So by sending an action potential in this direction, that stimulation can be passed on to these two neurons and to these four neurons, and can cause activity in these four muscle cells that I've drawn here. So in this way, using a diverging neural circuit, you can have multiple cells under the control of a single neuron. And this is really helpful in the case that your brain needs to coordinate a particular movement, let's say, that requires stimulation of a number of different muscle cells at the same time. So in order to effectively do that, you would want all of those muscle cells to be under the control of a single stimulus. So this is effectively managed through a diverging neural circuit, where you have just theoretically a single neuron controlling many cells, and that is made possible because of this diverging circuitry. So the opposite of a diverging circuit we'll see here in just a second. So the second type of neural circuit that we'll discuss here is a converging neural circuit. So instead of the signal starting with a single neuron and spreading out to many, here you have many neurons and their input is converging to a single neuron. So instead of spreading out, it's converging. So just for a practical example of this, many of the sensory neurons that allow you to perceive the world around you, they exist in these converging neural circuits. And that's because the signal eventually has to go to a specific place in the brain. And so it's useful to have numerous sensory inputs traveling into a smaller and smaller pool of neurons until it gets to the place where your brain can process it. So here in this example, you have a bunch of sensory neurons. Let's say that these are allowing you to have a sense of touch. So there could be thousands of sensory neurons that I'm activating right here, and their signal telling me that I'm feeling pressure on my arm is going to converge into a smaller pool of neurons that can then take that information to my brain to be processed. So this allows me to feel input from many different neurons, but they eventually converge, and as they travel toward the brain into a smaller, that signal is transmitted in a smaller and smaller number of neurons. So sensory neurons usually they tend to have this type of neural circuitry where their input signals converge and lead to a smaller number of neurons. The last thing I want to mention covering nervous tissue is the fact that it's not just neurons in the nervous system. There are also cells called neuroglial cells. And whether you're talking about the brain and the spinal cord, the central nervous system, or the peripheral nervous system, there's a different complement or a different set of neuroglial cells that are present in these regions. So in the central nervous system, you have oligodendrocytes, which form the myelin sheath. They wrap themselves around neurons, the axons of neurons, and increase the speed of transmission, and also prevent loss of the signal. So they insulate neurons. Astrocytes are going to regulate the chemical composition surrounding neurons. So neurons are very sensitive tissue type, and so they need a very tightly controlled, tightly regulated chemical environment around those neurons. and astrocytes help to do that. Astrocytes are called astrocytes because they kind of look like stars. So they also are going to anchor blood vessels and neurons in place. They kind of act as the glue. And they also repair neurons and form the blood-brain barrier. So the blood-brain barrier, again, is kind of a way to control the chemical environment inside the brain, to keep what's in the blood separate from what's in the brain. And so astrocytes help to do that as well. Microglial cells, even though they're called microglia, they are phagocytes. They are going to be swallowing harmful debris and potentially pathogens. So they're kind of the cleanup crew of nervous tissue in the central nervous system. And then finally you have ependymal cells, which are going to kind of line the cavities inside the brain and spinal cord. And some of them even secrete cerebrospinal fluid and pass this cerebrospinal fluid around. Cerebrospinal fluid is going to be kind of a nutrient and oxygen-rich fluid that bathes nervous tissue. So ependymal cells are kind of critical for circulating cerebrospinal fluid, or CSF. So now we'll move on to the peripheral nervous system. In the peripheral nervous system, the set of neuroglial cells is a little more simple. There are only two types. There are the Schwann cells, which, like the oligodendrocytes in the central nervous system, are going to provide a myelin sheath in the peripheral nervous system. And aside from the Schwann cells, you've got the satellite cells. So Schwann cells are kind of like oligodendrocytes, and satellite cells are called satellites because they surround and support the cell bodies. They can kind of be found around the cell bodies, kind of like they're in orbit around the cell bodies, like a satellite. But they're going to support the cell bodies of those neurons in the PNS. And like astrocytes, they're going to control the chemical environment around those cells. So again, neurons are highly sensitive to any changes in the chemical composition of their environment. And so satellite cells are going to play a support role by controlling it. So that'll do it for the nervous system. I know this has been a lot of information in a short amount of time, but I'll be posting a study guide soon, and hopefully these videos and that study guide will help with that information.